Maria is a medical student and research intern at the Center for Genomic Medicine at Mass General. She was born in Brazil. She was among the top 0.05% of the 45,000 applicants selected for a prestigious fellowship in Brazil, and is part of the largest leadership program in the country. She is now here in Dr. Patty Mussolino's lab. Patty, can you raise your hand for us? Just stepped out, okay. Patty's in the back here for those of you who have not met her. Take a chance to meet them both. So she's going to be conducting research on the investigation of plasma biomarkers and brain imaging for early cerebral ALD detection. And she's at her university, she established the first business academic partnership in medical research, and now hopes to transform the lives of people with cerebral ALD through translational medicine. So this, I should preface a little bit, the Emerging Investigators Award is for promising young investigators who are, we think, going to be future leaders in the world. This is us investing in them. This is funded by your philanthropy as a community, your gifts to ALD Connect to support future generations of leaders. So thank you very much for welcoming Maria. So.
Okay, hi everyone. It's an honor to be here. Thank you so much for the space. As Keith was saying, my name is Maria. I'm from Brazil. I work with Patty. And I'm going to be presenting to you our work on microvascular dysfunction in plasma and MRI as biomarkers of early CLD in response to treatments. So I wanted to start with this little boy here, Connor, who made history in the ALD field. Right when he was born, his newborn screening had him diagnosed with ALD, which shocked his family. And then a couple of months after that, he had early signs of CLD in his MRI scans, and that made his family move quickly to seek treatment. Connor was unfortunately not eligible for stem cell transplantation as he did not have donors, but thankfully he was eligible for the recently approved Skysona gene therapy that had just been approved a week before that. And then Connor received the gene therapy, and thankfully, a few months or almost 2 years after gene therapy, he's developing well and his disease progression has halted. However, this is not the case for the majority of CLD patients. We know that less than 20% of boys with CLD currently qualify or are eligible for the available treatments. And this is mainly due to lack of optimal donor, a delay in diagnosis, and limited access to expert centers. If we eventually focus on delay in diagnosis, we are able to give patients time to find donors, either find donors or find the expert centers that they need to go. So we think that this is the most strategic gap to tackle. As we know, current CLD diagnosis relies on early MRI lesion detection, but that exposes kids to anesthesia every 6 months, which we know is a super stressful process with sedation and contrast needed. And it also puts the patients at risk of missing the optimal therapeutic window due to a risk of late brain lesion detection. So are we able to detect childhood CLD before the MRI lesions take place? We know from previous work in our group that microvascular dysfunction precedes CLD lesion development, specifically in sensitive MRI images. As we can see here, lesion is seen only a few months after microvascular dysfunction, as we can see here, is already happening. This is a measure of blood-brain barrier permeability and capillary blood flow. So in this project, we have gathered the main ALD groups, and the idea was to characterize lesion progression, brain water diffusion, white matter integrity, and microvascular perfusion dynamics, correlate that with quanterics, angiogenesis, and neurodegeneration plasma biomarkers. So we know that microvascular dysfunction precedes blood-brain barrier disruption, which leads to lesion detection. When this happens, patients can have access to rescue treatment, which is the treatment that we already know, stem cell transplantation and gene therapy. We currently already have ways of detecting CLD non-invasively through NFL, but as a neurodegenerative biomarker, NFL can only detect CLD when brain injury is already present there. So what if we focus on blood-brain barrier disruption specifically? And if we try to find biomarkers that track blood-brain barrier disruption specifically, we will be able to predict MRI CLD lesion onset, opening up the preventive window of CLD treatment instead of just the rescue treatment, and we will be able to track response to treatments. So when blood-brain barrier disruption happens, the blood vessel damage that happens in the compensatory remodeling leads to a fluctuation of these main vascular biomarkers, which I'll be talking about here. Our data has mainly shown that there's no significant difference in when we compare healthy controls and ALD asymptomatic patients, and there's also no difference when we compare ALD asymptomatic patients with CLD patients. However, as we can see here, there's a trend of higher levels of angiopoietin-2, PLGF, and HBGF between healthy controls and ALD asymptomatic patients. Interestingly, when we look at the CLD group specifically, most of the vascular biomarkers that I just mentioned, they are positively correlated with each other in this group, except for angiopoietin-2 and PLGF. And then interestingly, once again, when we look at the gene therapy group specifically, we find significance because these three biomarkers, angiopoietin-2, PLGF, and HBGF, they are biomarkers that respond rapidly to treatment. This is a graph, pre-gene therapy and then post-gene therapy. We can see that their levels flip throughout time. And so the question now is, can the plasma vascular biomarkers that we are talking about here be used to assess response to treatment? We know that when blood-brain barrier disruption happens, there is a capillary flow disturbance in the blood-brain barrier, which makes the blood become the flow of the blood becomes heterogeneous instead of homogeneous. That alterates CTH, which is an MRI parameter. That leads to a diffusion dysfunction, which elevates the ADC MRI parameter, and that degrades the myelin, which makes the microstructure of the white matter gets disrupted. That decreases fractional isotropy, which is a measure of white matter microstructure, and decreases the metabolic demand in the brain, leading to the lesion detection. This is a little video so we can see what's happening in terms of capillary flow. Previous works have also shown that microvascular function, which is measured by these MRI parameters here, it actually does improve after gene therapy. And then what we have found here mainly is that angiopoietin-2, PLGF, and HBGF, which are the biomarkers that I'm talking about the most, these biomarkers are positively correlated with capillary blood and flow dysfunction in CLD patients. We can see in all of these three graphs here, these three biomarkers, they correlate with MRI microvascular dysfunction at baseline in CLD. So there's a potential of them being biomarkers of microvascular dysfunction. Then angiopoietin-2 and PLGF specifically, after gene therapy, we can see that there is the way that they behave is similar. Both of them show an early increase after gene therapy in terms of concentration, and they also correlate similarly with the normalization of capillary blood flow and capillary flow efficiency, which is measured by oxygen extraction fraction. And then these two biomarkers, angiopoietin-2 and PLGF, what happens is before gene therapy, they follow opposite patterns when we compare when we correlate their levels with capillary blood flow, sorry. And then after gene therapy, they follow the same pattern, which is similar to CTH, which is the exact capillary flow disruption and heterogeneity that we're talking about. So this happens in less than 200 days. So they are rapid representatives of response to treatment in CLD, which is in contrast, which you can say is a contrast with NFL, which is the biomarker that we were talking about, because as we can see here, NFL takes more than a year to normalize its levels after treatment. Other works have also indicated angiopoietin-2 as a sensitive marker of blood-brain barrier dysfunction in other neurodegenerative processes. So in Alzheimer's disease, there is that correlation between angiopoietin-2 and blood-brain barrier dysfunction, which is interesting in our analysis of angiopoietin-2 for this work. And finally, HbGF, this biomarker levels, it mirrors the metabolic demand, which is measured by, this is brain metabolism mainly. We can see that there's a really similar pattern between biomarker level concentrations in the treated group and the analysis of blood metabolism throughout time. So to conclude, what we can say that microvascular dysfunction in the brain measured by perfusion imaging is actually an early marker of CLD lesion onset and response to treatment. We can also say that the plasma levels of angiopoietin-2, PLGF, and HbGF, which are the biomarkers that I'm talking about here, they highly correlate with white matter microvascular dysfunction in CLD. We can also say that the changes in HbGF plasma levels, they mimic the white matter cerebral metabolism dysfunction over time in CLD. And then these same three biomarkers, they are early, dynamic, and sensitive markers of response to treatment, which make them surrogates of blood-brain barrier disruption. And while it's not significant in our small cohort, we can also say that these vascular plasma biomarkers, they might be able to detect early changes in blood-brain barrier that precede lesion onset when we look at the MRIs. So the final question that I want to make here is, could these markers that we're talking about here, could they be followed longitudinally and serve potentially as earlier predictors of brain lesion onset in our ALD patients? Our ultimate hope is that we can say yes to that question and that we're able to multiply the hope that Connor's story carries. This is a phrase that I wanted to end with. This is Connor's mom phrase, and she goes, "Connor is living proof that when it seems like everything is going wrong, everything else can go right." So this is our hope. And with that, I'd like to express my deepest gratitude to all of you. I'm honored to be here once again. Thank you for the invitation. All of this work would not be possible without all of these people here that helped us. And so I'd like to acknowledge them all. And thank you for your attention, and I'm happy to take any questions.
Thanks. We might take, let's take one question.
That was really great. I remember meeting you last year, and I think you're still incredible as I met you last year. I'm over here.
Oh, thank you.
Yeah, question for you on the blood-brain barrier leakiness. Do you have a sense how soon that happens before a lesion forms?
Yeah, one of the images that we had, we can see that those blood-brain barrier, so the blood-brain barrier markers, the capillary flow dysfunction and blood-brain barrier permeability, 10 to 15 months before the initial contrast enhancement happens in the brain, we can see that happening. So I would say this is not exact, but this is an idea of what we currently have with the studies we conducted.
Great. We're going to move on to applause for Maria, please. Yeah. Our next speaker, Nikki Coveñas, and I'm going to let Stefan Kemp introduce Nikki. Thanks.
Okay, so Nikki Coveñas, he started 3 years ago in the group of Mark Engelen and Vivi Heine. She's a stem cell expert. And me. So he has three PIs. And I still remember the first day we met 3 years ago. We were drinking coffee, and we have this potential PhD student in front of us with three people who have a long history in research. And Nicky was just sitting there, and he said, I want to work on microglia. And we were all very happy because microglia is the target for this disease. It's also the cell line, the cells that we have no idea, or we lack a lot of information about this cell. So finding, or yeah, it's not finding because you found us, I think. Having a PhD student who is fully dedicated on microglia is I think phenomenal for all of us because his research will tell us and learn us a lot about these cells. So yeah, I will stop talking now and.
Okay. Thank you very much, Stefan, for this nice introduction. So my name is Nicky Kovina. I'm a PhD student over at the Amsterdam UMC. And the title of my presentation is actually not the title of the Emerging Investigator Grant, but it's a model I've been working on for quite a while and that I've also used as part of the Emerging Investigator Grant. So first of all, who am I? I'm a PhD student over at Amsterdam UMC, and in my research I use stem cell models to study adrenoleukodystrophy, and I focus specifically on the role of microglia to try to understand how the microglia are affected and why or how they play a role in ALD. So my project, as Stefan said, is a collaboration between different groups. It's between the ALD group over at the Amsterdam UMC and also between the stem cell lab over at the Vrije University of Amsterdam, where I do most of my stem cell cultures. So I thought I wanted to make this presentation as simple as possible, so hopefully everybody can understand. So I'll start with the first thing, which I think most people know, but what are stem cells? So stem cells, simply said, are cells that are capable of becoming any other type of cell. And when we talk about iPSCs, or induced pluripotent stem cells, we are talking about adult cells, such as fibroblasts or blood cells, that we reprogram into their stem cell state. And these induced pluripotent stem cells we can then differentiate and transform into any other cell type in the human body while keeping a patient-specific genetic background in these cells. Very important, what are microglia? Simply said, they're specialized immune cells in the brain that are important for scanning the microenvironment, and they perform essential functions for nervous system health. They have several functions. One of them, a very important one in ALD, obviously, is that they support and they maintain myelin, which is the isolating layer around neurons, which are the brain cells that are actually communicating with each other. The microglia are also important for regulating the growth of brain cells and the differentiation. They're also very important for building and refining connections between these signaling brain cells that we call neurons. They survey the brain for harmful substances, including viruses, bacteria, and they also remove unwanted cells and cellular debris. And why do we actually want to look at microglia in ALD? Well, I think by now most people know, but they're a very high expressor of the ABCD1 protein, which obviously is affected in ALD. And from some people in this room, we know that they've done research where they have shown that microglia are affected early on in ALD pathology. And from other fields, we also know that when microglia are accumulating lipids, which is also occurring in ALD, that they can start to dysfunction. And maybe most importantly, we also know that they play a very important role in the therapeutic effect of hematopoietic stem cell therapy or transplantation. And because I don't have that much time, I couldn't go into all the details about the actual model that I made, but I do have a poster, so if you're interested, you can view my poster or come to me and ask questions. And one thing that I did want to show about the microglia model is how we actually make them in the lab. So we start out with our iPSCs that we culture and we expand them, we make sure we have enough, and then we dissociate them, put them together again, and allow them to form these clumps of cells, which we call embryoid bodies. When we grow these embryoid bodies, they start to form these bubble-like structures, which eventually attach to the culture plate. They start to spread out, and at some point, these, what I call factories, start to produce the microglia precursor cells, which we then take into another plate, add factors, and fully mature into functional and adult microglia. So, and I do that every week. I collect, once you have a factory going, you can collect them every week and you have to plate them and use them. So it's a very intensive culture, but it's also very fun. And now for the actual investigator grant. So I proposed to investigate neuronal networks using multi-electrode arrays on these iPSC-derived neuron microglia co-cultures. And first, I'll explain what are multi-electrode arrays. So multi-electrode arrays are culture plates that have electrodes embedded into the bottom of the plate that allow us to measure neuronal activity that is generated by cells like neurons. And there are different metrics that we can measure. We can measure single spikes, so single firing of neurons. We can also detect burst activity when there are multiple neurons next to each other are firing. And we can also detect network bursts, which are synchronous firing of multiple electrodes across the whole plate, which is basically a measure for a fully functional neuronal network. But I didn't say what neuronal network actually are, and simply said they're interconnected neurons that communicate with each other, which is important for normal brain function. And the reason why we want to look at neuronal networks is because we've actually, in the stem cell lab, we've also shown that microglia are very important for the formation and maturation of these neuronal networks. And we know that ALD microglia are affected. And also in ALD pathology, we see a concurrent loss of axons and myelin, which indicates that the neurons are also affected. So therefore, we wanted to look if ALD microglia could affect the formation and functioning of these neuronal networks. So that was the first research question, and the second research question was to see if we can distinguish cerebral ALD cells or neurons from AMN cells by using these multi-electrode array measurements. So implications of this proposal was that we would get a better mechanistic insight into ALD pathology. We could potentially identify neuron intrinsic effects or microglia-dependent mechanisms. And the ideal situation would be that we could actually develop a functional readout of ALD in vitro that could detect differences between cerebral ALD and AMN for early phenotype prediction. So the experimental setup, we made iPSCs, or we had iPSCs made from controls from patients that had cerebral ALD involvement and also from AMN patients. And I made neurons from these and also microglia, which I put together with the support of a red astrocyte layer to allow the neurons to fully mature and be fully functional. And then for quite a lot of weeks, I was measuring these cultures, and alongside I also had multiple time points where I performed immunocytochemistry so that we could actually image the cells and see how they were looking and what was actually happening. And the experiment, there were two main experiments in this project, or in this grant. The first one was to look at the effect of ALD microglia on control neurons. And the second experiment was to look at the effect between different phenotypes. So we had control neurons with control microglia, cerebral neurons with cerebral microglia, etc. And in the first experiment, when we look at the Mayer measurements, we actually do not see any differences. So it seems that the ALD microglia did not differentially affect the formation and functioning of these neuronal networks. And also, when we did immunocytochemistry to look at the morphology of both the neurons, the microglia, we also looked at synapse counts, the amount of neurons, etc., but we did not find any differences. And while we did find many different morphologies for the microglia, there was no clear significant difference between either the phenotypes or ALD compared to control in general. But in the second experiment, we looked at the complete phenotype, so matching neurons and microglia. And there, the normal spike rate and the burst rate, so regular activity, did not really differ. But when we look at the network burst, it seemed that in ALD neurons, there's either a delay of this formation and also there's a less network activity. But the interesting thing is, when we looked at the cultures, we actually saw very clearly that in the cerebral ALD neuron cultures that there were less neurons, they were less complex, but despite this, they still have a similar activity pattern, which is puzzling and could mean that maybe the cerebral ALD neurons are either firing too much and something is happening to them. But unfortunately, I did only complete this experiment last Monday. I was still imaging at the microscope, so I haven't been able to fully analyze everything. So for now, some preliminary conclusions. In the first experiment, we do not see that ALD microglia affect the control neurons in the setup that we are using. And in the second experiment, we see that neuronal network activity is reduced in the ALD genetic background. And specifically in cerebral ALD neuron cultures, we see that there is a reduced complexity and also fewer neurons. But despite this, there's a similar amount of activity, just not in network, in a network form. And the next steps that I'll be working on for the coming weeks is to complete all the quantifications, where we'll be looking at the morphology from the second experiment. I'll look at cell survival for the neurons, the microglia. I'll also look at the subtypes of the neurons that are actually present. I'll look at the synapses and the ratio thereof. And I also did one last test at the last week that I could still perform. And there I added C26 lyso-PC to see what elevated C26 lyso-PC would have an effect on all 3 different cultures. And with this, I would like to end my presentation and thank everybody that has helped, and also thank everybody that's here. And I'm happy to take questions. Yeah.
Great talk, Nikki. Questions for Nikki? Yep.
This is great work. Congratulations. Just a quick question: the attempt to count the number of neurons, is it in the same vials as your MEA vials, or is it different? Because that could play a role as to.
Sorry, can you repeat the question?
So you counted the number of neurons in cerebral ALD, and you showed that based on the picture, it looked like it was lower.
Yeah.
But the MEA activity was the same, the electrical activity. Was that done in the same vial, in the same valves, or was it different? Because there's so much variability in terms of confluency of the cells and so on.
It was not done in the same well, because in the MEA plates we actually cannot perform the stainings and also not do the imaging. So alongside the MEAs we had the imaging plates where we plated the same density, same amount of cells to quantify. But as I said, I haven't been able to fully quantify, but based on all the pictures that I've scanned through, it seems very clear that there are less neurons present. So yeah.
One more question.
Yeah, hi, thanks, excellent talk. 2 questions quickly. On those last slides, are they, firstly, all fibroblast or all PBMC-derived iPSCs?
These iPSCs that we have, they were generated a while ago, and I think they were made from fibroblasts. But now I think we've changed mostly to PBMCs. Yeah. But all of these were made from fibroblasts.
Cool. Yeah. And second part of the question is the differences you see between the CALD-derived and your AMN or control. Do you have any thoughts what could underlie them, and any approaches of how you want to look at that? I'm thinking like epigenetics, question mark.
So I could see how, obviously, with it, if you look at AMN and cerebral ALD, there's involvement in the brain, and the neurons that we made were actually cortical neurons, so they're neurons that are present in the brain. So I did expect that if we would see an effect, that this would be in cerebral ALD. But for now, after I've done all the quantifications, then I can get a better insight of what actually is happening. Because it could be that, for instance, the interneuron populations, which are usually more susceptible to cell death or stressful environments, that they are actually the ones that are lost. But for now, I cannot make any statements about it. Yeah. Okay.
Thanks very much, Nikki. That was excellent.
Yeah. Thank you.